All right. Hello, everyone. My name's Christine. I'm a PhD candidate at University of Technology, Sydney, based in the Future Reefs Lab. And today I'm going to be telling you about some work in collaboration with Coral Nurture Program, which you heard a little bit about from Paige yesterday. And it's looking at the use of a high-throughput phenotyping device to guide coral stock selection. Firstly, I would just like to acknowledge and pay my respects to the traditional custodians of the land and sea on which I live and work, particularly the Kuku-Yalanji and Irukandji traditional owners, and I pay my respects to their elders past, present, and emerging. So as I'm sure we're all very aware, being at the conference this week, coral reefs worldwide are facing significant declines, which is prompting a range of active interventions to help combat coral loss and maintain reef function and diversity. Selection of corals with beneficial phenotypes is one potential strategy to increase reef resilience through active interventions. And a phenotype is the observable characteristic of an individual resulting from the interaction of their genotype with the environment. Now, corals with identified advantageous traits such as heat or disease tolerance has the potential to bolster reef resilience through increasing the biomass of strong performers within the population, as well as amplifying the abundance of stress-resilient genes within the gene pool. Now, historically, coral phenotyping has been undertaken through laboratory studies, which makes it quite costly and inaccessible to reef stakeholders who require on-ground or in-water applications. However, over the past few years, we have seen the development of phenotyping devices for in-field assessment of coral phenotypes. An example of this would be the coral bleaching automated stress system, which is also known as CBAS. Recently, our lab group at UTS, at Future Reefs, have developed a high-throughput multi-taxa phenotyping device, which I'm just going to refer to from now on as the MTP device, which allows for rapid in-field assessment of coral phenotypes under multiple stressors. So this includes factors such as temperature, light, and nutrients. Now, similar to other phenotyping devices, the MTP device measures active chlorophyll A fluorescence and uses the response of photosystem 2 as a broader holobiont response to stress. And the reason for this is twofold. So firstly, because the majority of coral metabolic requirements are fulfilled through primary production through their algal symbionts, and secondly, because the coral symbiont breakdown is a sign of metabolic stress that manifests itself in chlorophyll A fluorescent signatures. Now, other phenotyping devices, such as CBAS, have successfully resolved both between and within species differences in coral phenotypes using this technique. Therefore, that's really demonstrating its value for restoration purposes. So we wanted to test the ability of the MTP device to do this, but particularly so in the field where it would be applicable to reef stakeholders. So that brings me to the three objectives of this work. Firstly was to investigate the ability of the MTP device to resolve coral physiological performance across a diversity of sites. Secondly was to examine variations in population thermotolerance thresholds and ranges as a means to potentially identify more tolerant populations or individuals. And a third objective of this study was also to assess how phenotypic diversity and genetic structure align. So for example, do we find sites that have a higher phenotypic diversity also have a greater genetic diversity? And this is important because approaches which select corals based solely on their phenotypic traits have the potential to reduce genetic diversity by selecting a subset of the population. However, we know that genetic diversity is also critical for maintaining reef resilience. Therefore, we wanted to understand the patterns between phenotypic and genetic diversity to understand if this approach of selecting more tolerant individuals could have an impact on the overall genetic diversity of a site. So to do this work, we collected 184 acropora and millipora fragments from 3 reefs and 12 sites across the continental shelf of the Great Barrier Reef, just north of here near Port Douglas. And we selected these sites because their cross-shelf location promotes a diversity of environmental conditions. So firstly, we have Lowell's Reef. It's an inshore reef located about 15 kilometers from the coast, a platform reef which surrounds a sand and a mangrove island. And due to its nearshore location, it's characterized by high summer maximum temperatures and greater temperature variability throughout the year. Now, in addition to this, corals growing within the mangrove lagoon at Lowell's Reef are also subject to more extreme conditions, including a higher temperature, reduced pH, and reduced oxygen. Next, we have Tongue Reef. It's a large mid-shelf reef, which is characterized by high summer maximum temperatures and more moderate temperature fluctuations. And lastly, we have Opal Reef, a offshore reef which is characterized by more stable annual temperatures, most likely due to support from oceanic influence due to its outer shelf location. So fragments were collected from acropora millipora colonies for phenotyping conducted on the vessel using the MTP device, as well as for later genetic analysis. So to tell you a little bit more about the MTP device itself, so it actually combines 3 existing instruments. So it uses a PAM imaging fluorometer capable of measuring chlorophyll A fluorescence, 4 broad spectrum white LED lights, and a thermocycler which provides a temperature gradient. 6 fragments from each coral colony were placed side by side in a custom well plate, which you can see in that top right photo. They were covered with ambient seawater as well as glass to prevent any bubbles from forming, and then that was placed onto the thermocycler, which you can see in the bottom right photo. So once the corals were on the thermocycler, they were subject to a 3-step protocol. So stage 1 consisted of a low light adapted baseline at ambient temperature, which in this case was 29 degrees Celsius for 10 minutes. Stage 2 consisted of a temperature ramping, ramping of the temperature gradient, across the 6 wells, with temperatures ranging from 24.5 to 37 degrees Celsius. And this was under that same low light condition. Now, once the corals reached temperature, they were then subject to a 15 stepwise interval of increasing light over a 7.5-minute period. Lastly, stage 3 consisted of a temperature ramp down and a return to that baseline light condition for a recovery period. Chlorophyll A fluorescence was measured during stage 1 and stage 2, as you can see from the T1 and the T2 in this figure, and that was to assess the response of the corals both prior to and during stress. For the genetic analysis, we collected genomic DNA from those same acropora millipora colonies and sent samples to diversity arrays technology for genome complexity reduction-based sequencing. After a series of quality control and filtering steps, we were left with about 2,000 SNPs, or genetic markers, and these were analyzed via a structure analysis and principal component analysis to allow us to assess and visualize population structure across the 12 groups that I mentioned. All right, so a little bit about what we found. So firstly, to assess the thermotolerance of coral colonies to acute heat stress, we used the ED50 of the relative maximum electron transport rate, which is the RETR max you can see in the y-axis of this figure. Now, ED50 represents the temperature at which each colony reached 50% of its initial RETR max value, and it has been used in a range of studies in the past to characterize the thermal tolerance of coral populations as well as individuals, but particularly in sea bass experiments. So as a hypothetical example, the ED50 of this colony would be 37.3 degrees Celsius. So this is showing you the mean ED50 as well as the variation in ED50 across the 12 habitats for the 3 reefs, Opal, Tongue, and Lowell's reefs. So we found that there was a significant difference in ED50 between the groups, with with Tongue and Opal Reef having the strongest performance. Now, in contrast, we found that Lowell's Reef had a significantly lower ED50, particularly the corals growing on Lowell's Reef flat, which is that top group there that you can see, as they were actually already showing signs of visual stress when we collected these colonies. In addition to having the lowest ED50, the Low-Isles Reef flat corals also had the greatest variability in performance of about 4 degrees Celsius in ED50 between the poorest and the strongest performer. When we looked at the top 10% of colonies with the strongest performance, or the highest ED50, we found that they were spread amongst habitats and reefs across the continental shelf, as you can see from the red dot in this figure, sorry. So this suggests that individuals with potentially greater thermal tolerance are well distributed throughout the system, irrespective of the environmental conditions. Moving on to the results from the genetic analysis. So this is a figure here showing you a principal component analysis, and it's showing that there's significant clustering amongst the 12 habitats with no discernible population groupings. So this allowed us to infer that all 12 groups belong to a single interconnected population. Now, alongside measures of population structure, we also looked at genetic diversity to assess the richness of the gene pool for each of the subpopulations, and we found that genetic diversity was fairly similar across the 12 groups, with the exception of Low-Isles Reef Flat, which had a significantly higher genetic diversity compared to all other groups. So when we think back to that third objective I mentioned, where we wanted to look at how patterns in phenotypic and genetic diversity align, this figure here is showing you the larger the circle, the larger the value for the trait, either ED50 or genetic diversity. So firstly, given that all 12 habitats belong to a single population, we determined that phenotypic diversity was not driven by the underlying genetic structure. Secondly, you can see for the most part that patterns in genetic diversity don't really match patterns in ED50 values. With the exception of Low-Isles Reef Flat, the top group there, we did find that it had the highest variability in phenotypic performance and also the greatest genetic diversity, which could indicate that variations in the thermal tolerance of the corals at this site are driven by the greater richness of the gene pool in this subpopulation. So just to finish with the implications of this work, so firstly, we found that the MTP device complements existing phenotypic devices and provides a viable means of resolving differences in phenotypic performance of acropore and millipore corals across diverse sites. The presence of naturally stronger colonies scattered throughout the population is an encouraging finding for bolstering population resilience through potentially through strategies of selection and propagation at a local scale. We found that phenotypic diversity did not align with genetic structure, and this suggests that proactive local management aimed at the selection and propagation of more resilient colonies should have little impact on genetic diversity within this region. Finally, the use of the MTP device provides a low-cost strategy for practitioners to select stock, which is immediately implementable and could help enhance reef resilience. And with that, I just wanted to say a big thank you to funders as well as to Wavelength Reef Cruises, who provided a lot of in-kind support for this project. So thank you.
Thank you, Christine. Wonderful work. Do we have any questions for Christine?
Just a quickie, Christine.
Sure.
So if I bought you some coral samples, how long would it take start to finish to get that analysis done on the boat?
Yeah, thanks. I probably should have mentioned that earlier when I say high throughput and rapid, what that really means. So with the MTP device, we can measure about 4 genotypes or 4 colonies of corals in about 40 minutes. Yeah.
You're right.
Thanks so much, Christine. That was really stress, but then also, sorry, were you able to hear that?
Yeah, I did.
Okay.
Yeah, thanks.
And then some potentially longer-term heat stress experiments, how those might
differ?
Yeah, that's a really good question. So with this study here in particular, we wanted to kind of validate the MTP device within the field, but the device has been developed by somebody else in our lab group who has been doing a lot of sort of comparison and validation studies alongside CBAS in the lab to really get to those questions. And so far, he has found that for the most part, the results are very similar between CBAS and the MTP device. In terms of the sort of longer term heat stress, I would say that there's, I think, always challenges there in sort of looking at chronic versus acute heat stress and whether they line up or not. I think there needs to be some more studies done to really check on that. Yeah.
Thank you, Christine. Round of applause, please.